Hi, everyone, and welcome back to our Computational Biology Reading Group series. Today, since this is the first series of the new year, we are going to do something slightly different. So today we'll be joined by Matthew Hershey, and we will be discussing the role of serendipity in research. And we'll specifically be looking at two examples of how we both came across code that was applied in a different context. And we realized that with a little bit of tweaking, we could use that code to answer our specific questions of interest. So we'll go through the details of that. And the second thing that we want to touch on is this kind of what next problem. Like the thing about languages like R or Python, they're quite high level, and the basics are actually pretty, they're not particularly difficult. It's quite easy to learn how to make a basic plot or do some simple data wrangling. But then when you move on, when you see what the experts can do, it's difficult to know how to bridge that gap. And one way to do it is to just see what code people are writing, like follow people who are doing things you admire on Twitter or star their GitHub pages and things like that. And so today we'll be covering two examples. Both of them have to do with plots. So we'll also be going over some ggplot code. Yeah, and with that, Matt, please go ahead.
Hey, Akshay, good. Thanks for having me. I'm happy to be the guest today and not the host as we've done in the past. Before we dive into the code, I wanted to ask you, um, what was your motivation behind having a session where we're devoted to serendipity and how we're applying some of the things that we see in other fields to our field? What got you interested in this, or how did it start?
So my main motivation was so serendipity, fortunately, can play a role in every area in life and other parts of research. Like for example, you might be reading a paper in an unrelated field and you come across a method that you think, oh, this would be perfect for my project. The problem with that, though, is it takes weeks, months, or maybe even years for that to bear fruit. But the great thing about in silico research is if you do come across that perfect code snippet, it takes like an afternoon to solve your problem. And that's something, again, you can't plan for that. But when you do come across that perfect code snippet, then applying it is pretty straightforward. So that's kind of what I wanted to touch on.
Yeah. How do you keep them organized, right? If we're gathering information from all sorts of places, you mentioned starring GitHub pages and these sorts of things, but what does your folder of good ideas look like?
I think this is where you can give better advice than I can because I end up just sending myself emails and just like trying to keep my head above water. So I don't think I have any useful advice. I'm still learning.
Yeah, no, sending yourself emails is an interesting solution. I don't think I would go with it though. So, yeah, I mean, I think I like that you're doing this, and I think one of the most interesting parts is to consider pulling ideas from a lot of different places. Steve Jobs famously said, "Creativity is connecting things." So when we come up with a creative solution to a visualization or to an approach to solve a problem, on one hand, we're just doing something that somebody else did. But when we take it and we apply it to our field, it ends up being a creative approach. And I quite like that. So,
Yeah, no, that's nicely put. And just to add to that, one of the great things about coming across this code snippet that solves your problem is, at least for me, it makes me realize that there are lots of things that I don't really understand or assumed I understand and understood how to do. But by applying it to my problem of interest, I really am forced to see what each line of code is doing. And the funny thing is, what I'm about to show today, I am revisiting this after like 2 years. So there was a few lines I had to look up the help documentation because I completely forgotten. So this is also sure it solves your problem, but again, it's another opportunity to learn.
all right. Well, thanks for doing this. So, I will show an example first. And so we'll just go ahead and dive right into the code. And so, Akshay, before we do that, you said that did you have a chance to put this code into the CBRG package so that the students can get it, or should they wait until after? What's the best way here?
So I think the best thing is for you to go ahead and go through your code. But yes, I have put both your code and mine, and I'll go through that after you go through your code.
All right. Thank
So
you.
maybe for now, I think what people can do is just watch, and this is always just I think better to watch first and then afterwards go through the code.
Okay, so let's dive in. All right, so the project that I'm showing you today is an example project that I have created for the introduction to data science, which Akshay and I developed and called Tidy Biology. So in that class, we have the students who are going through to put together a final project. And the final project is essentially a data set, a little bit of exploratory data analysis, and then a single simple visualization that captures some element in that data set. And to give the students a flavor of what one possible final project could look like, I put together a little toy example. And that's the example I want to show you today, because that example was inspired by a friend and collaborator of ours, Cedric Scher, who has a almost now iconic blog post called The Evolution of a ggplot. And I'll show you that in just a moment. But let's go ahead and walk through some of the code. The first thing that we did is we needed a data set. And this data set was one from the, let me just get it here. So this data set is one that we used for this class, which was a small table that I scraped from Wikipedia. I'm sorry, Wikipedia, that has information about the human chromosomes. And so here's just a setup chunk of some of the packages that I'm using, the tidyverse, and then many of these packages, ggRepel, ggforce, are going to be for visualization. And then finally, this one is going to be the package that we made for our Tidy Biology course, appropriately called Tidy Biology. So what I wanted to do first then is to get this data. And so rather than showing a bunch of code to scrape the data from Wikipedia, what we then did is we put this as a data package within Tidy Biology. And so I can simply run a data chromosome. And when I do this, it loads it now into my environment up here in the upper right. And then I can view chromosome if we want to just take a quick peek at this. And here what we have is an ID. This is a character a variable because we have our X and our Y chromosomes. So the ID is the number of the chromosome. And this is the length of the chromosome in base pairs, variations, protein coding genes, pseudogenes, several elements of our long non-coding RNAs, microRNAs, ribosomal RNAs, and so on, and then also the centromere position. And so this is essentially a architectural snapshot of the organization of the human chromosomes. And so what we wanted to do then is we take this and we wanted to get a little bit more information. The first thing though, before I show you what this does, I wanted to just take a glimpse at this data frame. And so glimpse is a great little function where we can glimpse chromosome. And when we run that, we can see that we've got 24 observations and 16 different variables that I just ran through for you here. So I wanted to explore the relationship between the number of protein coding genes and the number of base pairs. And when we do that, we can run this chunk. And when we do that, what we can see is that, not surprising, longer chromosomes have more protein coding genes. So when we compare the number of base pairs to the number of protein coding genes, now if we stop for a moment, we say, well, this makes sense. This makes sense given that if you have a longer chromosome, the number of genes that you could fit on that chromosome would be greater. And that's generally what's true here. So chromosome number 1 is the longest, and chromosome number 22 is among the shortest. Now, when some of the very early studies on karyotyping and visualizing the human chromosomes, it was interesting. There was actually a side note here. There's a big debate very early on whether or not humans had 23 or 24 unique chromosomes. And it was assumed for the longest time that it was 24 because chimpanzees have 24. And so people would look and they say, oh, well, I only count 23, but I must be missing one. And so for the longest time, this dogma persisted. Well, it turns out that on, I believe, chromosome 2, there's a fusion. Our human chromosome 2 is a fusion event of 2 chromosomes that are in chimps. So interesting. So anyhow, so these are generally numbered from longest to shortest, 1 all the way to 22. And then X and Y, of course, are our sex chromosomes. So we see this, and that's not surprising. Okay, well, we want to do a little bit of more exploratory data analysis. This got me thinking about variation, and so I was curious, what the amount of variation of the chromosomes looked like. So we're going to go back up, and what I wanted to do then is I wanted to take my chromosome data frame, and I wanted to make a new column here that was called percent variation. And this then is going to be the amount of variation divided by the number of base pairs. And so we're adding this as a new column, and we can then come down here, and we can then start to look at the amount of variation that is within each human chromosome. So in order to do this, let's just run this and see what it looks like. So in order to do this, we have the number of variations, and these are going to then be differences in our DNA sequences from the reference sequence. So what's the amount of variation that's going to be generally in our DNA, and how is that related to the length of the chromosome? So again, not surprising, there's this pretty tight relationship between the length of our chromosome and the number of variations. In fact, if we look at this relationship here and we compare it to our base pair and our protein coding gene relationship, we can see this is a much tighter relationship. But I noticed two things immediately, is that the X and Y chromosomes have much less variation than the rest of the chromosomes. So I thought that was interesting. And I wanted to think of a better visualization, a better way to depict that X and Y appear to have less variation. Well, this is where serendipity comes into play. And so let me show you this first. This is the blog post that I told you about by Cedric Scher. And if we go up to the top, we can see that this is called Evolution of a ggplot, part 1. And this is a great blog post. If you haven't seen it yet, you can go ahead and just Google Evolution of a ggplot, and you can put in Cedric's name, and this will come up. And what he does is he takes you through this start of the ggplot, and these are then just snapshots along the way of how you can make this ggplot better. And I thought one of the most interesting things and choices that Cedric made here, and so this is looking at the student to teacher ratio of different countries grouped by continent to then infer something or learn something about the number of students to teachers across the world. And I thought one of the most interesting decisions here, and you can see the exploratory data analysis that Cedric is doing of thinking about different ways to plot these data. And the decision that he makes is to put these lollipop plot lines, and this comes down a little bit further. Sorry if I'm making you seasick here. But what you can do is you can then compare your student to teacher ratio to an average. That's going to be important because we want to know, for this example, whether Europe, North America, South America are high or low. But of course, high or low is compared to what? And so you can then compare this to the global average. And then one way to visualize this easily is to connect the mean of a continent compared to the global mean and come up with a very quick visualization of, is this high or is this low? And then I was able to learn about this annotate and adding some of these arrows. And so there's a couple of different ways that you can add some labels. And be forewarned, as soon as you start doing this to your ggplots, you'll spend a lot of time fiddling. That's a technical term. In order to get the plots and the labels and arrows just right. And so after a lot of fiddling, Cedric gets this. And finally, he displays his final plot. And what you can see is here this mean of each continent of students per teacher compared to the global mean. And then, of course, he puts the raw data underneath. So now, if we go back to the example that I was showing, what I wanted to do was show whether or not there's a lot of variation or a little variation compared to something. And so I figured out that X and Y chromosomes are clearly lower, but how to visualize this. So I chose this lollipop method and some of the code from Cedric's blog post to visualize the differences in the variation compared to the mean variation. And so if I run this code, you can see that I have mean variation and percent %var doesn't exist, so we'll just run the whole thing. I don't think I ran that top part properly. So we can just run this whole thing. And what we end up with then is this is my final plot where we can see that sex chromosomes have lower genomic variation than other chromosomes. So what I wanted to do was show the average genomic variation, and that's this line here. So a little bit like Cedric, I can drop this vertical line. I then annotate it because I have the amount of variation here, but annotation allows you to draw the viewer's attention to a very specific part of the graph, right? Because there's a lot of things going on here. And when I look at the graph, if I stop and think about it for a little while, I can certainly take in all of the information. But you, as you're making a visualization, you want to prioritize the information that the viewer is seeing when they are looking at this. And so I wanted to point out that the average genomic variation was 4.9. I wanted to then plot these chromosomes, and I put the chromosome number in the lollipop so that people could very easily see it. That means I also don't need another legend. It also means that I can take, instead of having chromosome the name over here and asking people to then draw a visual, an imaginary line with their eye, I can say, okay, here's chromosome 1, and they can infer that these are the rest of the chromosomes. And then I can use this GG highlight, and I think GG force is also a package that I use here, where I can call out some specific points on here, and I can then, again, the main take home is 3 points. First is that X and Y are lower than the rest of these points. How much lower? Well, I can annotate with the numbers. I then also say lower compared to what, and that's the average genomic variation. So this was the example. And then for the students, I say, here's the conclusions. And then I also, for the students, we discuss some prioritized follow-up studies, because a lot of data science in the biological sciences is going to be used for hypothesis generation. How can we use these data to come up with a hypothesis that we then take forward into the lab? And because this is such an important part for the students, I wanted to ensure that this was also a part of the project, is to have them answer the question, well, what are the experiments that you would do to follow up on the interesting observation that you made from here? And so here, I would say maybe chromatin states are known to influence genomic variation. Therefore, I could look at the chromatin state of these genomic loci, including histone modifications or DNA methylation. And then, of course, I make some acknowledgments. And so if I knit the whole thing together, we end up with this nice report, summary report of the data set, the early exploratory data analysis, and the visualization. And so this then, just as Akshay and I were talking about, I happened to read the evolution of a ggplot around the same time that I was preparing the materials for the first iteration of this course. And so it was serendipity that I read that resonated with me, and that I was then able to apply that to the project. And of course, a big shout out goes to Cedric for doing some of the heavy lifting on getting the code that allowed me to get started with this. But this is just one example where we can collect bits of information and types of visualizations, and probably most importantly, approaches to visualizing something or approaches to solving a problem. And we want to then have this wide net that we cast to capture these ideas, so that these are then at our disposal when we need to solve some problem. So that's the example that I wanted to show you today. And Akshay, I'd be happy to talk about it more, or if there's any questions that you want to ask, or perhaps we can, if there's questions, go ahead and light them up in the comments. But that was the example I wanted to share today of one way I've applied serendipity to make what I think is a more effective visualization for a data set.
Great, thanks, Matt. Yeah, I wanted to say that this kind of plot definitely has that wow factor, and I really I've seen so many of your students when they see this plot for the first time, it really gives them motivation to learn more about R and ggplot because they can see what it can do for them. I was going to ask this question, but I think you already answered it. You mentioned that you were thinking of a way to, an effective way of illustrating, coming up with a visualization for these data, and then around about the same time you came across Cedric Shear's blog post. Were you actively looking for inspiration just on the internet, or was it just like you stumbled across a blog post and you realized, oh, I can apply this to my
Oh,
problem?
yeah, in this case, I wasn't actively looking. I didn't have this idea of how do I make a better plot or how do I evolve a ggplot. In fact, one of the things that I was participating in around, I guess, 2018, 2019, was just when Tidy Tuesday was getting started. And Tidy Tuesday, of course, as you know, is the global activity of visualizing a data set that is released on Tuesdays that then there goes your Tuesday and Wednesday and Thursday working on visualizations. But the fun thing is to see the different approaches that people will take to their Tidy Tuesday plots. And so I was doing Tidy Tuesdays, and then I was seeing some of the things Cedric was doing and some of the others in the community. And I thought there's another example when there's a Tidy Tuesday data set, a very early one, that was comparing wages that are reported in Australia between men and women of the same profession. And this was a way to compare, two states across many, many variables. And I thought that this like level setting line, rather than just saying which is high and which is low, to say compared to what, I thought was another example that I was able to apply to this project. So it's these things that sort of stick with you, and when you understand the approach and you understand the thinking that went in behind that, if you agree with that thinking and agree with that approach, then you can take that and you can apply it to a lot of different types of similar problems.
Thanks for that. And thank you for bringing up Tidy Tuesday because that serves as a nice segue to my portion because I too, like my plot was inspired by a Tidy Tuesday plot. And I'm not a particularly active user of Twitter, I'm more of a lurker. But one thing that I've learned is following people who not only produce great code, but are also generous enough to share it. So I think that's really wonderful. Like, I think that would be the number one piece of advice I'd give to anyone who wants to become better at coding. Just follow the right people on Twitter. And you also mentioned this thing, the importance of fiddling, technical term, to get your plot just right. And that's another great thing when you see, an example of, for example, a plot that you really like, that person has already done a lot of the hard work of just like working out exactly like how big the font, like a particular label is supposed to be, or the curvature of a line, things like that. So that's also it's a nice benefit. All right. Thanks again, Matt. I guess I'll move on to my portion.
All right, what are you going to show us?
So I am also going to show a plot that I saw, and this time it's by another person who is very well known in the R plotting area, Georgios Karamanis. Um, and so it was the same thing. This was when I was working on a project in Jim White's lab. Jim White is a PI at the DMPI. And it had to do with phosphoproteomics data. And this was looking at the phosphoproteome of the effect of long-term caloric restriction on the muscle phosphoproteome. And those data, phosphoproteomics data, were generated by Paul Grimsrud, who is a regular participant in the CBRG meetups. And I wanted to come up with an effective way of illustrating the connections between kinases and substrates in my data set. Something that was qualitative, but would immediately draw the reader's attention to the important kinases that came up in the study. And I just couldn't work out a way of doing it. So I was like, I'm just going to put this in as a table. And then I came across this one plot that I'll show by Georgios Karamanis. Again, just like you, around about the same time I needed a solution, I saw this and it clicked. And adapting it to my code I basically was almost just copy pasting. So yeah, I'll go through the details of that.
All right, let's see what you got.
Cool. All right. So I am going to share my, oops, just give me a second. Okay, I'm going to toggle back and forth between our studio and a web browser. But before we actually go through the code, let's once again just reinstall the CBRG package. So we do remotes colon install GitHub Duke cct/cbrg. CBRG is in all caps. And you remember to set force equals true so that it overwrites the package if you already have it installed on your machine. And also, as you can see, that I've set up a project associated with these meetups, CBRG underscore meetups. It's great if you do that. It's not required though. So hopefully the installation, or rather reinstallation, should not cause any problems. And once you've installed it, just load the CBRG package library CBRG, and you can use one of the functions, launch project, to launch the required, the necessary Markdown document. And so this is meetup number 10. So we're already into double digits, which is great. So we do launch underscore project 10. And with any luck, this should now appear on your screen. And once again, let's just go ahead and save as, just so that when you do, if you do make any changes, it will be reflected here. So I'm just going to do this and save it as 10 underscore project. And you should see it come up in your project directory. And so let's go ahead and run the setup chunk. So these will install or any packages that are required for all the packages that are required for this walkthrough. And you will notice that one of those packages is a package that Matt has made called Teddy Biology. And this contains data that he used to make his plot. So if you look at your console, R will ask you for a few questions. So the first question is, would you like to update any packages? So again, it's a good idea. If one of the choices is to update all of them, just update all of them. And that should hopefully just take a few seconds. While that is running, or actually, I think I'll just wait for this to run. Okay, so all good. It was able to install and load all the packages that we need for today. Matt also mentioned a couple of packages that I don't think we've specifically gone over, like ggRepel and ggforce. And those are add-on packages to ggplot2 that allow you to do certain things that ggplot2 itself doesn't allow you to do. And so once we load all of that, if we go back to the code, you can see that this R Markdown file has been divided into two sections. One is on line 55, is simply called chromosome plot, and this is Matt's plot. And to kind of see the layout of the R Markdown file, you can go up to this little button here that says Show Document. And if you click on it, you can see that we have it's like a table of contents here. So if we click on chromosome plot, we see Matt's code, and Matt has various subsections where he describes both what the code is doing. So this is really great. So you can see lots of helpful comments that show you what each line of code is doing. So this is something really useful to go through to make sure that you understand what the code itself is doing. But Matt has also included text here that conveys his reasoning process. So that's all of Matt's code here, and I've run it previously, and it should run fine. And then if you go click on kinase substrate here, so it'll take you to line 182, and this is the plot that I will be discussing. So as I mentioned to Matt, this is I had a phosphoproteomics data set, and so in that data set were lots of phosphorylated residues and fold changes that were associated with the effect of caloric restriction. So it's caloric restriction versus ad lib. And I what I did is, so you can imagine that this was a big data set, kind of like a RNA-seq set or a microarray set. So you can look at specific residues and try to work out what the underlying biology is, or you could sort of take a 1,000-foot view of all of your phosphorylated residues and try to work out what kinases, or infer what kinases might be involved in this phosphorylation pattern. And there's this really nice approach called KSEA, kinase substrate enrichment analysis, that allows you to do just that. I won't really go into the details of it, but briefly, what KSEA does is, actually, what I'm going to do is I'm just going to stop sharing this, and I will just go through the KSEA process. So the people behind KSEA have made a Shiny app, and I'm just going to reload this. And the good thing is they also have a sample data set, which is just a sample of phosphoproteomics data set. And so I'm just going to upload that quickly. Apologies for this. Right. And then if we look at the output of this, what we see is, if I just run KSEA, let me just do this again. So just bear with me. So we have sample data set, choose for upload, upload complete, click go, great. So this is the output that you get from KSEA. So it shows you that, I haven't shown you the underlying data set, but based on the phosphorylation patterns and the phosphorylated residues, KSEA produces a plot like this. And this is comparing two like treatment groups. So in this case, it's uh, samples that were treated with a drug compared to samples that were treated with a vehicle. And what this plot is meant to show is, what it does is it works out a score for each of these kinases shown here. And what the score tells you is, it's kind of an aggregate representation of the effects of your treatment on the target residues of these individual kinases. So for example, this kinase up here, CSNK2A1, this one is red. So that means it's significant. It's red and has a positive number. That means it shows you that it suggests this kinase is activated by the treatment. And conversely, the ones that are blue here, it shows that these kinases are suppressed by the treatment. So it's a pretty simple output. It's quite intuitive to understand. And I was able to easily produce a plot like this for my data just by using this tool. What this tool also shows you is it gives you a table of kinase substrate links. So this shows both the kinase and the substrates that the specific kinase targets. Unfortunately, there's no way to visualize this, or no built-in way to visualize this. And I thought it would be nice to be able to qualitatively show the connections between the kinases and their targets in my data set. One reason is to just see what the identities of the targets are. So for example, if we go back to this plot, you can see which kinases are being actively affected by your treatment, but you don't know what the underlying target sites are. Whereas with the kinase substrate links, it shows you explicitly that this ABL1 is targeting tyrosine 95, and there's this associated log fold change. So I wanted to just show the connection between the kinases and the specific targets. I also wanted to see if there was an overlap. So for example, are there multiple kinases targeting the same residue? And that might be an important thing. So I couldn't work out a way of doing this until I came across this beautiful plot by Georgios Karamanis. And this is kind of where the serendipity aspect comes into play, because this plot has nothing to do with phosphoproteomics or caloric restriction or even biology. What this plot is meant to illustrate is it shows you it's built using data for Nobel Prize winners. And this is a data set, a Tidy Tuesday data set that was released in, I think, 2019, around about the same time I wanted to be able to plot this data. these data. And this data set contains information on where Nobel Prize winners were born and where they died. And this plot illustrates, so on the y-axis, you can see all the countries. So these are the countries of birth. And on the x-axis are countries of death. And these individual lines represent a specific Nobel Prize winner. And what I find both impressive about this beautiful plot and slightly depressing is if you look at the y-axis, you can see that there's a pretty even distribution of Nobel Prize winners based on country of birth, suggesting, clearly indicating that there's no monopoly on talent. Talent is evenly distributed. But if you look at where they all end up, almost all of them end up in the US. So anyway, so my idea was really simple. I took Georgios Karamanis' code, and what I decided to do was I decided to make the x-axis, instead of country of death, I made it the kinase or kinases, and the y-axis were the residues. And so that way you can identify links between residues and kinases, and also see things like, is there an overrepresentation of a particular kinase? For example, is one kinase associated with a bunch of residues? And you can visualize things like overlap. All right, so that is all the stuff about kind of the inspiration. Now let's just quickly go through the code. So if we go down again to line 182, that's where all of this starts. And here's a link for the plot. And I've also put in a couple of links to other truly wonderful plots made by Georgios Karamanis. So if they can serve as inspiration to you, that'll be wonderful. So if we go to line 192 and just run this, we import the data. If we just click on it, we see what it looks like. So this is the output, typical output of the KSEA application. So you can see that the first column has the UniProt IDs of the various kinases. The second column has the names of the target genes, and the third column has the modified substrates, the target residues. Oh, and it also has this information on what database is being queried, like where are they getting this information from? And they use this website called Phosphosite Plus. And each residue is associated with a certain log2 fold change. So this reflects the effect of caloric restriction in this specific example. All right, so that's what the raw data look like. The first thing I did was, so if we go back to the data, you can see that columns 2 and 3 are separate, but it would be nice to be able to just combine that. That's useful information. You have the name of the gene followed by the name of the target residue. So to do that, to combine two columns using in R, you can just, you take your data frame and then you use this function called unite. And unite takes in, so the first thing you supply is the name of the new column you want to make. You give the names of the columns you want to combine, and then you can put in a separator. So, once you combine the two columns, how do you want to separate the information in these two columns? You can use a comma, you can use whatever you want. So in this case, I just used a space. So if I run this and create a new data frame called cr_v_al_ptm, you can see that now the columns 2 and 3 have been collapsed into just one column called ptm. Okay, and that's kind of all you have to do. And your data set is now ready to be used. And you can now use Georgios Karamanis' code to reproduce his plot. So I haven't, it's not especially clear here, but you can see that there are quite a few kinases in this data set. I want me to actually quickly get a sense of how many kinases there are. You can just go to the console and type in unique CR and the name of the data frame, and then $sign and the name of the column. And you can see that there are 20 something kinases here. And that's really busy for this plot, at least I found. So what I did was I decided to only focus on the 7 kinases that had the most number of targets. And to do that, I've put in a question here: what are the 7 most quote popular kinases? I'm just leaving that as an exercise to the people who are watching this. It's a pretty simple solution, but that's just there. So anyway, the answer to that is the 7 most popular kinases are these kinases here on line 207. Um, and oh, I should say, actually, when I said that this data frame is ready to be plotted, that wasn't entirely true. We need to do one more thing before it's ready. And that is, what we need to do is, for these 7 kinases, we need to decide what color. Um, so in the Karamanis plot that I showed you, the connection between the residues and the kinases, they each have a different color depending on, I believe, depending on what the country of death was. So in this case here, you want, for example, this kinase here, P05132, all the lines that connect to it, you want them all to be a specific color. So that way, just looking at the connections, the color of the connections, you can work out what the associated kinase is. So
Hey,
to do that,
Akshay,
yes,
just
question.
one quick question from the chat. So we've got Tim asking, are the thickness of the lines providing some quantitative information as well?
That's a wonderful question. The answer is no. And now, in hindsight, I think it would have been a lot more effective to have the thickness be proportional to the log fold change. But that's a really nice question. And I'm not brave enough to try to, on the fly, work out how to do that here, but that would be a nice exercise. But yeah, thank you for that. So yes, the thickness of all the lines in this particular visualization are the same. Um, and so the first thing we need to do is, so one thing with this code here is, this is an example of how you can chain together lines of dplyr code and ggplot code just as one contiguous kind of mass of code. Um, that it's completely up to you if you want to do it like that, or if you would prefer making an intermediate data frame and then using that data frame to pipe into the ggplot code. In this case, I've just put everything in one big line, which can get a little bit confusing. So let's go through this. We start off with our data frame, and then we pipe it into this filter function. And since I just want to choose these 7 kinases, I can use a filter function for that. Notice, though, that I'm not using the equals to sign that we're now familiar with. That is if you want to just filter those rows that have, for example, just uh, one kinase. So if I wanted to just have a resulting data frame that had rows associated with this kinase highlighted here, I would just do equals to equals to. However, if there are more than one kinase, if there's more than one kinase, then you have to use this slightly strange looking in operator. So it's percentage in percentage. That's the only thing that's a little bit strange. So when you have multiple things that you want to filter on like this, you should use this in operator. So actually, one thing that I've shown previously, but I think is worth showing again, is when you have multiple lines of code chained like this, especially if it's someone else's code and you want to see what each line is doing, what you can do is at the end of a given pipe, you can just type in view, capital V, and then open close parentheses. And if you run that, so if you're on a Mac, if you do Command Enter, you'll see what the intermediate data frame looks like. So in this case, and you'll see that there's a little dot, and that's because this data frame is not associated with any object that exists in your environment. It's just something that you're creating on the fly. So if you compare this intermediate data frame with the original one, you'll see that the original one has 72 entries, but this one has only 44. So this kind of tells you that your filtering process actually had an effect on the data frame. You must remember to remove that view if you then want to see what the next line of code is doing. So that's that's what the filter does. The second bit, this bit is also, I think, is new, is that we to make those, to give colors, to assign colors to each of those curves, we need to, or Jojo's did was he explicitly defined what the colors of those connections were, I believe. Maybe he didn't, but I decided that I wanted to give each kinase its own color, and these are the colors that I, for whatever reason, decided on. So to do that, what you can do is you can specifically create a color column in your data frame. And you do it like this. So as you know, to create a new column, you can use the mutate function. So we use the mutate function. And then within the mutate function, you first give the name of the new column you want to make. So I'm just calling it color, which makes sense, equals to, and this here I think is new. We're using this function called case when, and it's quite an intuitive function. So let's just go through kind of what this code means. So the way you read this is, so case when, if you look at the first line within case when, it's asking, is the entry, if the entry is, kinase.gene is equal to this specific kinase, tilde, what it means is I want you to, in this color column, put in this value that's on the right of this tilde. And this value is a hex code. And it's one way of representing color in art. So what I'm saying here is this first line within the case when function, what it does is whenever, for all the entries in your data frame that have this specific kinase, the corresponding entry in the new color column will contain, will be this hex code. And so then we go kind of kinase by kinase, assigning separate colors. So if we then go to the next line, we see for this kinase, P2044, I want you to put the value red in the color column. And then you go to the next one, you do the same thing. I want you to put the value, whatever this hex code is, in the color column that corresponds to this kinase. And you do it like that until you have, until you've assigned a color, a different color, to each of your, in this case, 7 kinases. So to see what the effect of this mutate and case when all of this code is, again, you can go to line 218, and after the pipe symbol, again, just type in view. You see there's a new intermediate data frame. And this intermediate data frame now has a color column. And you can see how, for example, this kinase P05132, all the rows that have this kinase also have this one color. I don't know off the top of my head what this hex code means, but it corresponds to one color. And similarly, if we go down to the next kinase, P2044, all of its entries are associated with red and pink and green and so on. So again, if this is the first time you're seeing mutate and case when used like this, it can be a little bit confusing. But hopefully, if you compare this intermediate data frame before and after you run this mutate code, it should hopefully be clear what all of these lines are doing. Okay, so essentially, we've now taken our original data frame, applied this filter and these filter and mutate functions to finally make the data frame that we can now use to make our plot. So now we get to the fun stuff of the plot making. So there's an impressive amount of code here, but the majority of it, or at least half of it, has to do with the aesthetics. So let's first go through the plot building part. So as you know, to make a plot using ggplot, the first thing you do is you pipe your data frame into ggplot. So that's just on line 219. So you have ggplot. And then remember, if you want to map data from your data frame to a plot, you have to put all of those things, the things that you want to map, within this AES, this aesthetic function. So we do ggplot and then AES. And this is the part where it might get a little bit confusing because this is also new. So far, all the examples we've seen of ggplot have involved just an x-axis variable and a y-axis variable. So for example, a bar chart or a scatter plot. Here, we have two additional parameters. In addition to x and y, we also have this thing called x end and y end. And the reason for that is because of the geom we're using. And so far, the geoms we've used are like geom point, geom call, geom hist. We're using a new geom called Geom Curve. And actually, I just noticed when Matt was going through his code, he too used Geom Curve, but for a different purpose. He used it to make, I believe, his curved arrows. We're going to use Geom Curve here to make the curved connections between our substrates and the kinases. And Geom Curve, if you just go to the console, type in !geom underscore curve, you look at the help documentation, you see that yes. So if you go all the way down to aesthetics, you can see that this tells you what the various parameters of geom curve are. So you see there's this x, y, x end, y end. So if we read this, what this means is that we need to give geom curve two pieces of information. One is what are the coordinates of the start of the curve, and what are the coordinates of the end of the curve? And then what Geom Curve will do is just draw a curve between those 2 points. So here we have our starting point of x is equal to 0. So that's just, x equals to just basically the y-axis where it intersects the x-axis. And then the y-axis coordinates are your PTMs, or what I'm calling is PTM column. These are the target residues. The only thing is here, what Georgios did was he converted this PTM column into a factor. And once you've converted that variable into a factor, you can use one of these functions that is from this package called forcats, Forcats. And he uses a specific function called fct underscore rev. And the purpose of this line is to ensure that the residues, they appear in alphabetical order descending. So you start off, if something is like, if there's an AKT S39 or something, that'll appear at the top. And if there is some target protein that starts with a Z, that'll appear at the bottom. So those are your start side coordinates, starting coordinates. And the end coordinates, so the curve then ends, on the x-axis it ends with the associated kinase, and the y-axis is a fixed value. Now this could also be 0. That's like kind of the equivalent of the x-axis of the x equals 0 here for the target residue. But I think Georgios, for aesthetic reasons, chose 1. And then finally, you want to map the colors that you just created here to the individual curves. And to do that, you just specify color equals color. That looks a little bit confusing because the color here is the name of one of the parameters in Geom Curve that you can see in the help section here. And the color on the right-hand side of the equal sign is the name of the column that you created. So you can give it a different name if you, I don't know, you could do like my color, and then so that means here you would also specify my color just to differentiate the two. I'm not going to do that here because it might break my code. So that's the ggplot and the geom curve. And another thing the geom curve takes in is this curvature. And again, Matt brought up the importance of fiddling with values. This curvature equals minus 0.4. I can't honestly tell you that is necessarily the best curvature. I believe it's the one that Jojo's chose. So I chose that too. So what we can do at this point is, so another thing is, once we do this, after this, so lines 227 and 228, this, honestly, I don't, okay, let me just run this code here up to 226 as is. So again, what you can do is you can just pipe this, well, actually, you can just run it like this. And you see that ggplot throws an error. It says discrete values supplied to continuous scale. So to get rid of that problem, what you can do is you can supply these two functions, scale underscore x underscore discrete, and then scale underscore y underscore discrete. So that's kind of my explanation for why those two functions are there. And so if we run those up to line 228, we get this plot. And this is actually pretty close to what we want to get. And so in the last 4 minutes, all I'm going to say is the rest of this code here has to do with cleaning up the aesthetics. So you can see a lot. So we are familiar with theme underscore minimal. So this is one way of just quickly altering the aesthetics of our plot in general. We've come across this labs function. This is a way of applying labels to your axes and a title. This is where it gets a little bit tricky, this theme function, because the theme function does a lot of things. It can be used to change pretty much every aspect of your plot. And you can see that Georgios has done the hard work of making all of these different, minor adjustments here and there. So to sort of give you a quick, so let me just run all of this to show you what the final plot looks like. And that's this. So this connects the various kinases to their target substrate. So you can see that there are some kinases with lots of targets and some targets that I think are associated with, some targets that are associated with two or more kinases. Now, if you want to see what this theme function does, one really useful thing to do is to just comment out each of the different pieces of code that Georgios has included. So if I comment out line 235, this panel.grid, and then I run my code, I can see what that line has done. It's not very clear here, but if you don't have this line here, this line 235, you can see the ggplot puts in a very faint background grid of horizontal and vertical lines. And so when you do set panel.grid is equal to element underscore blank, you get rid of that background. So it's just an aesthetic choice. And so I will leave it to you, given that we only have 2 minutes left, to go through each of these lines, each of these pieces of code line by line, commenting them out, changing some of these parameter set, these arguments, and then looking at the resulting impact on your plot. And with that, I'm done. So Matt, any final thoughts, questions?
No, I thought it was it's a pretty heavy data visualization day, but I think the take home that we want to impress upon you all today is just that you can look at other people's code, you can learn from their code, and if you're lucky, you can accelerate your own project by applying code snippets. And that's one of the best things about this R ecosystem is code's available. People want to share it. People want to help other people and help you save time. So that's one of the things that I enjoy the most.
Thanks, Matt. So I think that's pretty much all. Thank you for showing, going through your walkthrough, and thanks for attending. And so next week, we're going to go back to the fun stuff of proteomics and Q features and S3 objects and all of that.
All right.
So, thanks, Matt, and thanks everyone else. Thanks for watching.
All right, thank you.